has to gather, and the cost of that part of the regulation is strictly proportional to the risk hypothesis, because only the process is based on a very particular and single risk hypothesis. When it comes to GMO, or genetically modified organisms, there is already a fixed shopping list of risk hypotheses. Sometimes those risk hypotheses are not very relevant, sorry, because they don't have much to do with the trait of the GMO. But nevertheless, in the regulation, it is established that the developer must gather information in regards to those possibilities, and therefore that the regulatory pathway is much more expensive and lengthy. Now we're going to see an example of these two kinds of products. Now on the left, you have an example of a new variety being developed by the exposure to radioactive substances. Of course, the exposure to radioactive substances only occurs in the laboratory, and the seeds that come out of that experiment are not radioactive. And of course, the descendants of that seeds doesn't have anything to do with radiation. But in the past, radiation was used to damage the DNA randomly. We do not exactly where, but it created new traits. In this case, they could select a trait for improving cowpea to be drought tolerant. And now the International Atomic Energy and the United Nations Agency for Food and Agriculture are helping Zambia to adopt this technology for the benefit of the farmers. Now on the right, sorry, you have an example where in a different continent, Bolivia, is seriously considered adopting a genetically modified soybean that is also drought resistant, but in this case, that was obtained by using or generating a GMO. This GMO was generated by an Argentine biotech company called Bioceres. So now, the idea here is we have, before genome editing appeared, we had two possible options. And then scientists began using a new tool in molecular biology, which are site-directed nucleases. And site-directed nucleases, the name means that these enzymes, these proteins, go to a specific place in the DNA that was selected by humans, and in that place of the DNA, they make a small damage, and from there you have many possibilities. So beginning from the left, if this site-direct nuclease just damages the DNA and nothing else is made by human intervention, the DNA repairs by itself, the cell repairs the DNA without any help, and you can have a mutation. So usually when we talk about genome editing, we talk about this simpler possibility. Then you have a slightly more complex possibility, number two, where the human intervention helps the cell to repair the gene in a predetermined way. So the human also dictates exactly how it's going to be made the repair. And in that case, we also are looking at a gene editing, but a slightly more advanced way of doing gene editing. Then we have number three. In number three, in addition to the use of the site-directed nuclease, a whole new piece of DNA with a whole new gene is introduced into the cell. And therefore, you have an introduction of a foreign gene in a precise location in the genome. So now we are seeing again a genetically modified organism or a transgenic organism. The only difference is now, instead of inserting this gene randomly, it is inserted in a very specific location. But for almost everyone, this is clearly a GMO. And then you have more complex possibility. In this case, I didn't put a graph showing how the modification is made in the DNA because it's very complex. So I put I included a graph on how the technique works. It's known as gene drives. The gene drives are used for introducing a gene in a population and helping that gene to spread faster than what usually would happen. So in this case, some people are describing this latest use of site-directed nuclease as a member of a wider group of new things called synthetic biology. So we can see here that using site-directed nucleases, we can have a whole range of different kinds of products or outcomes, and they are being designated with different denominations. But these are just genome editing, GMO, synthetic biology. So far are only words. The thing is, regulators need to decide what is the meaning of these words for legal and regulatory processes. So, for instance, in Argentina, we developed very early, we developed an approach in 2015. And our approach is based on the GMO definition for the Cartagena Protocol, which is the most widely recognized international agreement for the biosafety of genetically modified organisms, or as it is called in the protocol, living modified organisms. And that definition says that these kind of products must have a novel combination of genetic material and the use of modern biotechnology. And this in the end means the use of recombinant DNA. So on the base of this definition, we made a decision tree. We developed a decision tree in order to decide if a product obtained by these techniques is a GMO or not. And we not only develop a way of sorting between the two boxes, GM or GMO, but we also establish a way of minding the gap between the two regulatory options so they can cross-talk with each other and help each other regardless of the product being established to be a GMO or a non-GMO. So we accumulated in 4 years. Actually, we have been discussing this from 2012. We established the regulation in 2015, and during 4 years, we have been taking decisions on different products. So we accumulated enough experience as to classify many products obtained with different techniques in regards of those products being GMOs or non-GMOs. Since I have only talked about Argentina so far, this is a map I have borrowed from a colleague to show you what is going around the world about this. And we have many countries in Latin America that are having the same approach as Argentina nowadays. In North America, Canada, and the US have their own approaches based on a different rationale, but the outcome is very similar. And we have countries in Asia that are also having their own way of rationalizing this decision or regulatory decisions about GMOs, and for most products, their decisions are coincident with the ones of Latin American countries. And we also have developments in countries like Russia, China, and in the European Union. It is rare because in there you have a struggle between different outcomes. On one side, you have a legal outcome saying that all gene-edited organisms are GMOs because of a very special definition that they have in the European Union, which is not the definition of the Cartagena Protocol. But also in Europe, you have official advisory groups or other legal authorities or national regulators that said that genome-edited products, at least the simpler kind of genome-edited products, are not GMO. So in Europe, they are still struggling around this. And in this map of my colleague, there is nothing painted in Africa, but actually in Africa, you have countries like Kenya, Nigeria, South Africa, and others that are discussing about this, how to regulate this, and they have quite advanced drafts or quite advanced discussions on the regulation. So surely in an updated version of this map, you will see them participating too. So I'm about to finish, and these are like a summary of the ideas I have tried to convey to you. So basically that most regulatory frameworks in any country in the world already have the tools for handle genome-edited products, because in most countries in the world, they are already an established regulation for conventional varieties and GMOs. The only thing is to make a communication between them, to coordinate and to have clarifying regulations for these new technologies. But it's important that all countries try to establish their regulations about genome editing in the same way, that they are harmonized. It can be done because we share a very common base of scientific knowledge. So in terms of regulatory expertise and knowledge about this technology, all the countries are on the same page, basically. But our main obstacle, as I mentioned it for Europe, for instance, is that there are some countries that have their own definition of what is a GMO or what is a regulated biotechnology product. And therefore, from those differences in the definitions, we have problems in sorting out specific products. In any case, there is an international rule included in international laws that says that the regulatory burden for sanitary purposes can be as important or as much as justified to assure safety. But once you have the basic or minimal requirements to assure safety of a product, you shouldn't keep on building an exaggerating burden of regulation, because that hinders the development of the country, the socioeconomic benefits derived from innovation. And also because in here, it's important to take this other into account. In here, we can have products with the same trait, the same characteristics, the same benefits, the same hypothetical benefits or hypothetical risk, but obtained with different technologies. But those technologies were applied very early in the lab, and they are not present in the final product, in the sense that the radiation is not still in the plants, or the genome edited plants are not being edited 5 generations later and cannot edit any other organism that eats them. So therefore, it's important that all of these technologies, even when the method to obtain them in the first place is different, are regulated very similarly if the end product is very similar. In here, there is a list of references that back my presentation. They were co-authored by me. That doesn't mean that you have to read my papers. This is I just suggesting them as a first step, so you can get to the references and read other authors as well. And if you don't have time to copy this slide, just go to Google Scholar and Google my name and genome editing, and you will find it. Thank you for your attention. Well, I think Rodora is perhaps occupied, so
Okay, I was unmuted. Thank you very much, Dr. Dr. Lema. Now we are moving forward. We have consumed a little more than the time. So we're going to move forward for the next poll. Can we see the next poll, please? All types of gene editing techniques produce GMOs. Is it true or false? Thank you very much. Let us now see the results. Is it now available? Okay, yes, correct. Not all types. Thank you very much. I think our audience is very knowledgeable. Let's move forward, and I'm now going to introduce Dr. Nina Gloriani. She is currently the chair of the Philippines Vaccine Expert Panel and a retired professor of medical microbiology and immunology, and the former dean of the College of Public Health, University of the Philippines, Manila. She was director of a Semiotropical Medicine Regional Center and the director of the Institute of Biotechnology and Molecular Biology of the National Institutes of Health. Dr. Gloriani conducted various basic and applied researches on hepatitis A, B, C, and D, HIV, herpes, and Hantan virus, and leptospirosis. Dr. Gloriani is a clinical microbiology consultant in the Institute of Pathology, St. Luke's Medical Center, Quezon City, and was the president of the Biotechnology Coalition of the Philippines for 6 years. She's a fellow of various medical and professional societies and continues to serve as the chairperson of the National Certification Committee for the Eradication of Poliomyelitis in the Philippines and a technical consultant to various agencies in the fields of medical microbiology, microbial immunology, and medical biotechnology. Let's have Dr. Gloriani on the floor, please. EJ, put on her there.
Oh, hello, everybody. I'm happy to
Ayan.
share with you the health applications in genome editing. So next slide, please. Jay. So this is the outline of my presentation. Just one slide for the beginnings and historical perspectives on genome editing for health. Then two of the strategies for gene editing. Then the main applications. I only have a few examples on two hematologic diseases, on cancer, and there are many applications in viral infections, but I will focus on HIV. There are many other applications, cardiovascular and metabolic and neurodegenerative diseases. Then I will summarize. Next slide, please. Well, I know Dr. Lema already presented the genome editing basics, but I just want maybe to emphasize as a microbiologist that all of these enzymes came from bacteria. So from E. coli, from Streptococcus pyogenes, and they're used by these bacteria to attack or destroy viruses. So it's like their way of dealing with invaders. So after all of these basics have been discussed, just to say that genome editing may be done in vitro or what we call ex vivo or in vivo when you directly insert into the host any of these targeted genes or any ablation, correction, and other genomic modifications. Next slide, please. So this is the in vivo strategy for CRISPR. Well, I just use CRISPR-Cas9 based gene therapy because it is the one that is most used in the health application. So by in vivo editing, we mean that the CRISPR-Cas9 components are directly delivered into the patient for in situ, so meaning there in that space and time, gene editing. They may use viruses to deliver the genes or non-viral vectors, which could be naked DNA, DNA encapsulating liposomes, compacted DNA, DNA nanoparticles, and other cell-penetrating peptides. So it is a direct injection into the host. In this case, human. We're talking about human. Maybe injected into animals as well. The ex vivo strategy actually does the modification outside of the host, in this case, the human host. So on the left side, you will see cell culture-based ex vivo editing. So these are adult somatic cells that will have to be taken from the host, and then in the laboratory, in vitro, they will have to be modified. So you see there, the cells will be expanded, clonally selected, and the correction will be made by the CRISPR-Cas9 enzyme system. On the other side, we have, it is possible to also change or modify pluripotential stem cells. I think this is the way to go for many of these health applications because you deal with the youngest cells of the hematopoietic system, for instance. Okay, next slide, please. Next slide, EJ. Okay. Now, for the examples, I think gene therapy for hematologic diseases have been with us for more than 5 decades already. But this CRISPR technology has introduced more precise modifications. And as example, we have, for instance, beta thalassemia. This is a blood-related genetic disorder which involves the entire absence of, or usually just errors in genes responsible for the production of hemoglobin. Hemoglobin is a protein present in the red blood cells which we use for oxygenation of our cells. So this is done ex vivo. The gene editing is done outside the body. And from the recent reports, CRISPR Therapeutics has edited more than 80% of human hematopoietic stem cells using their system to produce red blood cells with high levels of fetal hemoglobin. Fetal hemoglobin compared to adult hemoglobin can bind oxygen much better than adult form of hemoglobin. This is then transfused back to the patient. Next slide, please. So from beta thalassemia, well, hemophilia is really not very rare. Actually, we have cases of both hemophilia A and hemophilia B. As an example, I have here hemophilia B, which is an X-linked recessive monogenic disorder, meaning there's one gene that is involved. It affects 1 in 25,000 males. And these people with hemophilia B have mutations in the factor 9 coagulation component. So we have many blood clotting factors. So for hemophilia B, it's factor 9. These mutations will lead to spontaneous bleeding. So these patients or these people are not able to have their blood clotting. So in the past, actually up to now, the current clinical treatment of hemophilia B consists of lifelong therapy with injections of factor 8. So every time they will have to go through that injection. But with this gene therapy, we have CRISPR-Cas9 targeted integration of factor 9 gene using an adenovirus vector delivery. So much of what we can see or have been reported have been done in mice, but we hope to see some of this in the future in humans, tested in humans. Next slide, please. Now with cancer, I think this is where more people will be more aware or will be more interested. Now, there are 3 things I will have to share with you here. First is the concept of oncogenes. Cancer is generated by oncogenes. Actually, every person has what we call proto-oncogenes. These are not bad genes, but they can become bad, and we call them oncogenes. So oncogenes are bad genes that are permanently turned on or activated. So they're always on. So they produce this permanent activation allows them to grow out of control and lead to cancer. You know that if your cells continue on dividing, then that may lead to cancer. And some of these bad genes are due to mutations that activate these oncogenes by chromosome rearrangement, by gene duplication, or having extra copies of a gene, making too much of a certain protein that will make the cells grow unlimited. So the CRISPR system, what can it do? It can turn genes involved in cancer on and off. So for instance, our photo here, image here shows that CRISPR could potentially treat cancer by generating mutations through turning off the oncogenes. Actually, the same picture where they're both left or right show that the CRISPR system, the different enzymes, nucleases, can turn off these oncogenes. And the ultimate goal is to remove malignant mutations and replace them with normal DNA sequences. Next slide, please. So that is turning off. Now, we want something that the CRISPR will turn on. So also, any normal person will have tumor suppressor genes if we are normal. And one of the examples of tumor suppressor genes is P53. These genes will keep cells from dividing too quickly or slow down cell division and repair DNA mistakes. Now, there are family cancer syndromes where we showed or that been reported to show inherited abnormalities of tumor suppressor genes. So they have these mutations in their P53 gene that will allow cells to proliferate, cells to migrate, cells to invade, and cells to survive much longer than they should be surviving. So all that on the right side here, they're involved in cancer metabolism and in resisting chemotherapy. So CRISPR is able to turn on suppressor gene. So from the red, which is mutated P53, CRISPR could be used to turn on the suppressor gene and allow the patient to suppress the tumor or cancer. So the green one, we want the green one. Next slide, please. And well, those are turning on and off the genes. But we have another platform here, and this is actually being used in personalized medicine for cancer patients. This is what we call the CAR-T cell therapy. CAR-T cell therapy uses specially altered T cells. T cells are a part of our immune system in order to fight cancer, but T cells can also be used to fight other infections. So a patient's T cells are collected from the blood, then modified to produce special structures that we call chimeric antigen receptors. So they are a hybrid, chimera, on their surface. We may also call them CRISPR T cells. Now, these T cells are genetically modified to have cancer-recognizing receptors, which make them very efficient killer T cells, killing tumor cells. So this is currently an emerging field of immunotherapy for us in cancer or in oncology. So sometimes they use the term supercharging the patient's own immune system. So making the immune system of the host really charged up to fight cancer through genetic CRISPR was used to turn off PD gene, which is used by tumors to dampen immune responses against the tumor. Crippled genes, then the second is the crippled genes coding for the CRISPR would cripple genes coding for the natural T cell receptor. So you create a blank T cell, a T cell that will not recognize other antigens. And after creating that blank T cell, they will insert a designer receptor that will arm the T cell with cancer homing property. So this is the property that will make that T cell know which tumor antigen to bind to and which tumor to kill or to attack. So that is a very, very promising therapy, and actually there are many reports of that already on human trials. Next slide, please. So hematologic disorders, and we have cancer, and just one of the viral infections, because I worked with HIV before. So you know that HIV attacks or enters host cells, specifically those that are CD4 positive, like T cells, using certain receptors. Here we look at CXCR4 and CCR5. These are co-receptors for human immunodeficiency virus. So if that entry of the virus is successful, what will happen after several stages of viral replication, that the T cell will die. Now, how did they try to solve this using, of course, CRISPR? So they have identified, they use gene editing enzymes that have been prepackaged into adenovirus or a lentivirus. These enzymes could knock off or ablate or delete the CXCR4 and CCR5 genes. Of course, that they do that separately. So what you come up with is a letter C here, CCR5 and CXCR4 being deleted from T cell. So that T cell can no longer be invaded or attacked by the virus. So this has made the T cells in these patients resistant to further HIV entry and invasion. So next slide, please. The next slide will actually just show how this CCR5 gene editing has progressed over the years. So it started back in 1996 when they found that actually a certain person was found to have a 32 base pair deletion of the CCR5 receptor. And this person with that deletion was found to be resistant to HIV. And that is the one that gave them the idea that it is possible to edit this receptor. And over the years, now back in 2009 up to I think more recent ones, with clinical trials for this particular edited version of CCR5 or CXCR4 of T cells of patients with HIV has been ongoing. So next slide, please. Okay, so this is actually my last slide. So those only a few examples here, but we know that these health applications are increasing, but take note that most of these are largely personalized medical intervention for usually rare, complex, or challenging diseases like genetic diseases, mutation-driven malignancies, and viruses. Actually, aside from HIV, we have human papillomavirus, hepatitis B virus, and EBV. These last three viruses are actually oncogenic viruses, meaning they can also cause cancer. So a lot of CRISPR is ongoing also for their therapy. Now remember also that for humans, at least, the somatic cells and not the embryo cells are modified because we do have a lot of ethical issues. There has to be regulatory oversight. Of course, we're looking at cost and equity. Should these technologies be available to patients? We always look for safety, quality, and efficacy. What we remember that we need more improvements in technology so that we reduce off targets. You do not want to modify DNA unnecessarily. So you reduce off targets and at the same time, you increase efficiency in just targeting what you need to target. Now, just to say a few things more, I think microbes may be bad for us, but they help us in many ways because through their enzyme systems, we are able to do all of these genetic modifications for the sake of our patients. So thank you very much. That's all.
Okay, thank you very much, Dr. Gloriani. Those are very comprehensive slides, and the first time that I saw that really genome editing can work for our healthcare. Now we move forward and let's see the third, I think it's the third poll now. Can we see the third poll? Third poll question? Okay, all gene-edited products must be regulated as GMOs, true or false? Please press your button for true or false. And let's see the answers. Okay, that's correct. No, oh, yes, it should be false. Okay, now we're moving forward. I am looking at the many questions that come in for Dr. Lema and for Dr. Gloriani. Please let them come in. Let's move and look at the next speaker. Let's listen to the next speaker. Dr. Marcus Weiss is the strain director in Global Regulatory Affairs and Quality Management of DSM based in Switzerland. Royal DSM is a global purpose-led science-based company active in nutrition, health, and sustainable living. Dr. Weiss holds a PhD in cell biology and postdoc in the medical field. Dr. Weiss worked in the chemical and biotechnology industry, first with F. Hoffman LaRoche, and since 2003 was employed at DSM. He has held various managerial positions in R&D, technical product management, human resources, project, and program management. Ladies and gentlemen, let's welcome Dr. Weiss.
Many thanks, Margaret and Rodora, for giving me the opportunity to present in this webinar, and hello to everybody. It's a great pleasure for me to outline what impact genome editing has in industrial biotechnology. Let me start with a short introduction into our company. DSM is more than 100 years old and has started actually in coal mining. Over the decades, it has gone through many transformations. From coal mining, it went into fertilizers and petrochemistry, and from there into nutrition, health, and sustainable living. Out of our sales of roughly 10 billion annually, 2/3 go into animal and human nutrition and health, and roughly 1/3 goes into high-performance plastics. In the nutrition field, DSM is the #1 supplier of vitamins, nutritional lipids, such as omega-3 fatty acids, carotenoids, and other nutraceuticals, so products which can increase the nutritional profile of foods and feed. And often, these products are sold as pre-mixed custom nutrient premixes. For our ingredients, we use either chemical synthesis, extraction from biological materials, or biotechnology. The choice of the production method is often dictated by what is most economical, but also ecological. Those two aspects in industrial biotechnology are very closely linked. As you can see on the slide, sustainability is a core value of our company. We believe that technologies are not the solution, are not the cure for all the problems we have on our planet. But to solve those challenges that the world is facing without technologies is also not an option. In our view, genome editing has an important role to play. We've looked at the history on this slide. Let's also look at the history of genetic engineering, both from a technology perspective, but also from a regulatory perspective. 1973 marks the year of the first DNA cloning. And those who have lived the 1980s, they will remember the big impact that a single technology such as PCR can have to bring an entire field forward. And obviously the same happens again now with the genome editing techniques. At the same time, obviously there have been huge advances in DNA sequencing, from the first generation sequencing techniques that were also used to establish the Human Genome Project, to the second generation techniques which emerged as of 2004. Nowadays, it is possible quickly and at relatively low cost to easily sequence the entire genome of at least microorganisms. And this in turn allows to check that the genetic modifications we do with whatever technology, they happen as planned, that no unintended side effects are happening, or if such side effects are happening, what they are, and whether they have any impact on the safety of a microorganism. How does this now relate to the regulatory developments? In the 1970s, the DNA cloning caused a lot of concerns among scientists, and that's why the scientific community convened in 1975 in the famous Asilomar conference in the United States to define how to approach DNA cloning. They decided on a moratorium until a proper safety framework is in place. Shortly thereafter, the first NIH guidelines were published in the US, followed in 1906 by the Blue Book of the OECD. Still, in the logical sequence of the concerns of the early days, the first versions of the EU directives on GMOs were entering into force in 1990, and the same mentality also dictated the Cartagena Protocol on biosafety under the roof of the Convention on Biological Diversity. Why do I mention that, and why is it so important to emphasize it? While science and technology have continued to evolve and to make rapid progress, the regulatory framework is still grounded on the scientific knowledge and concerns of the early days and has not taken it into account adequately the scientific progress that has happened since then. Now let's move to industrial biotechnology. In industrial biotechnology, we use a relatively limited number of major workhorses to produce fermentatively a large number of products. Filamentous fungi such as Aspergillus niger, yeasts such as Saccharomyces cerevisiae, or bacteria such as Bacillus subtilis or Escherichia coli. As mentioned, a large variety of product classes can be produced fermentatively, and in many cases, genetic engineering has been used to improve either the process efficiency or to strengthen the safety of the production microorganism. Increases in process efficiency obviously typically result in higher product quality, in better economics, and in a better eco-footprint, so higher sustainability of fermentative production. Some product classes, however, particularly live microorganisms, they are still typical non-GM classes, not because there is not potential to improve also those microorganisms, but simply because of the public pushback on GMOs. As already presented in the previous talks, genome editing obviously is much more efficient, more precise, more targeted than traditional GM techniques. And as a result of that, in industrial biotechnology, use of genome editing is already now widely used and is the norm rather than the exception. I would even go a step further and boldly state that all products currently on the market derived from GMMs would now be developed using genome editing techniques, simply because it's much more straightforward. There are two important caveats though. Still, not all the microorganisms are equally amenable to genome editing, so there is still technological development needed. And secondly, even if the technology is there, companies may still not use genome editing at this stage because of the complex patent landscape and the associated legal uncertainties. Let's return to regulations, and we've heard already this distinction between GMOs versus non-GMOs. Although obviously the real world is not such binary to have only this distinction, the real world is much more gradual. As we've heard, the new genome editing techniques, they question this separation between GMOs and non-GMOs, simply because you can use them to introduce a single base substitution, which would be very much on this side. But you can also use these techniques to introduce entire heterologous genes, which according to the old definitions would be GMOs. Even worse, the EU Court of Justice has ruled in 2018 that plants and microorganisms obtained by genome editing, the newer techniques, are to be considered GMOs, which obviously has the result that products with the very same genetic changes and the very same properties are regulated differently simply because different techniques have been used to generate them. And then, as said before, for products that are still typically non-GM, obviously this ruling represents a strong disincentive against using genome editing for the improvement of such products, be it dairy cultures, probiotics, bread, wine, or beer yeasts, just as examples. In the interest of time, I will not go into detail on what these additional opportunities might be. I just refer to this article of Barangul and Notebart, which have highlighted that if society endorsed genome editing and would not link it to this stigmatizing GMO, non-GMO definitions or dichotomy, there would be a lot of additional opportunities for genome editing all along the food chain, making food safer, making food production more efficient, more sustainable. What does that all tell us? As said, first of all, genome editing is there. It is the rule rather than the exception in industrial biotechnology. As also outlined in the previous presentations, what truly counts are the benefits and properties of products, not how they are produced. The process-centric perspectives of the Cartagena Protocol and of many GMO regulatory frameworks, they are no longer fit for purpose. And now is the time to act, to redefine our approach to biotechnology so that innovative solutions can be used in the best interest of society and the environment. That there is hope. I have brought up a few recent examples. On one hand, the former EU Commissioner on Health and Food Safety, but also the German Minister of Food, Agriculture and Consumer Protection, which advocated for genome editing and that it should not receive the same destiny as GMOs in the past. Even more remarkably, the German fraction group of representatives of the German Green Party, Germany traditionally more against the GMOs, they have 9 days ago published a fantastic pamphlet for the benefit, for the use, the responsible use of genetic engineering overall, obviously including genome editing. And what remains is obviously that I count on all of us in this webinar that we also serve as ambassadors to bring those messages forward and hopefully to change the approach to biotechnology that we had over the last 30, 40 years. Thanks a lot.
Thank you very much. Thank you very much, Dr. Weiss. We're still waiting for a lot more questions, but while we do that, we can also put up our fourth poll, please. Okay, gene editing is more affordable than genetic modification. Okay, true or false? Thank you very much. Let's see the response. True, definitely true. Thank you very much, audience. Now we're moving forward to the question and answers. Can we have the gallery, please? Okay, so that we can have all our speakers on board. We're now going to, I have seen here some very interesting questions that every one of us will appreciate truly. So the first question would be from an anonymous attendee. In your opinion, is synthetic biology different from synthetic genomics? Anybody can answer. Unmute your microphones, please. maybe Dr. Lima. Is synthetic biology different from synthetic of genomics, or is it the same?
Sorry. Oh,
There.
this is the first
Unmute.
time I hear the term synthetic genomics.
Yeah,
People
this
is
first.
making up new terms
Okay.
all of the time, and this is not random. People usually coins new terms because you really have an innovation sometimes, but sometimes it's because they want things to be treated differently by society, regulated differently or perceived differently. So for me, in order to answer that, I should read a little bit more about the use of this synthetic genomic term. But
Okay.
nevertheless, in regards to regulation, you can make as many terms as you like. For instance, with GMO, people coined the term cisgenic instead of transgenic for certain applications, trying to have those applications treated differently. But in the end, what is important, the only thing important is the definition of a regulated article in your national law. You can make up as many technical terms as you want, but in the end, the only important thing will be the legal definition.
Okay. Thank you very much, Dr. Lema. There's another question for you here. So we'll just ask two questions for you and then there ask the others. How is the global, this is from Bambang Purwantara from our Indonesian Biotech Information Center. How is the global trend to regulate either genome editing or synthetic biology? Will they be categorized as GMO with very strict regulatory processes? How active is the National Biosafety Commission in Argentina to try to prepare this new technology accommodated into proper regulation?
Well, in Argentina, as I have described, we worked 8 years so far. We were one of the first national commissions in biology to work on the issue. And at least in regards to the rationale to regulate the products, we have things quite developed right now. So nowadays we are focusing on helping other countries on reaching the same status. In regards to what will be the global situation in the future, I cannot promise anything or be sure about anything, but the trend is quite good. At least when we began in 2015, we were one of the few countries in the world having a policy for this. And now, only 4 years later, many countries have policies that are very similar to the ones by Argentina. So I'm quite positive about the future.
Very good. Thank you very much, Dr. Lema. We have a question for Dr. Nina Gloriani. Maybe you know this. This is from Ryan Bedford of USDA PAS Manila. Where does the Philippines currently stand on genome editing regulations? Are they close to having a regulatory framework?
Well, I'm really sorry I cannot answer that, but as far as
Sorry.
I know, we are trying to address that issue from different agencies here. But I'm
Mhm.
sure there will be considerations for those. Well, if you're just talking about health, we have very stringent requirements for health products. But for other biotechnologies, I think the government is looking at that. All the interagency. Sorry, I cannot.
Okay, thank you. And then here's another one from Kenya, Archelius Anami. Uh, just an inquiry. If we knock out CXCR4 and CCR5 for HIV treatment, won't there be issues in the future? These receptors aren't useful in the first place, and will this be a treatment for both HIV 1 and 2 types?
I think we do not knock this out for all people. It's being knocked out for people who are infected with HIV. So as you know, HIV attacks mainly T cells. Of course, there are other cells like macrophages. So this is not to knock out everything because these receptors have some use for other things. So it's not just for HIV. So we cannot knock out everybody's receptors for that. I don't think it coming.
Okay, thank you very much. This would be a question for Dr. Marcos Weiss. From Jomerson Tolentino, what would be the effect now on our socio-economic and also in our environment? Is there a huge production of genetically modified organisms and products and genome edited products that can affect our socio-economic conditions and the environment in the pipeline, perhaps? Yeah.
Well, it all depends whether we make this distinction of what we release into the environment versus what we keep in fermenters and then properly dispose of at the end of fermentation. And also, obviously, it's a question what we call genetically modified, because genetic modification is something inherent to nature that happens anyway. And in my opinion, we overstate what human beings do in terms of the amount of genetic changes that we introduce and how different this is to what also naturally happens. But that's my personal opinion. So in that sense, the question is difficult to answer, because a genome edited plant or a genome edited microorganism could also happen in nature. Such single mutations, they are happening naturally and are there in much higher numbers than we can reasonably produce in industry. So again, it's not really helpful to look at the technologies and classify according to technologies, but really looking at what are the boundaries we have in our on our planet, on our society, the number of people we need to feed, and what solutions do we need to have to serve all the climate and biodiversity ambitions in the best way. And as said, we strongly believe technology must play a role in those solutions.
Yeah, that's a good answer. This question is, I think you can answer all this, because what is bothering people is missing the target during genome editing. Are there any mitigations or measures to correct all the missing targets or make it accurate somehow during genome editing?
Well,
Yes,
I again start with what is not considered genetic engineering. Typically, classical mutagenesis, as also addressed by Martin, is introducing many more changes that we do actually not know for. This is accelerated mutation. And there nobody cares about all those unintended effects. The beauty of genome editing is the much higher precision, that you know what you are doing, and that it happens at much higher precision. And what I said is that we should not only look at the technique of genetic engineering, but also on the analytical side, what we can nowadays do. And we can do for microorganisms already whole genome sequencing, so we can absolutely at the base level check what has happened in addition to the intended change, and then decide, is that really of concern or not? But due to the higher precision of the technologies anyway, there's much less of those unintended side effects to start with. And technology develops so quickly, this will also become the standard in higher eukaryotes over time. I'm convinced.
Right, that's correct. Do you have any additions, Dr. Gloriani or Dr. Lemma, on the targeting some leaky stuff during genome editing? How do we mitigate it? Well, Dr. Weiss has already responded very well. Now, there's one question here. Some of the people nowadays think that the GMO gene editing, gene modification has a negative effects on our body system, immune system, the rise of new diseases, or effects on the environment. How can we be assured that the new organism has no bad effects once it will be introduced to us or to the environment? Or so I mean, I think it's risk assessment, and
maybe I should. It's all right. Go ahead.
Yes.
Dr.
Please,
Lemma,
Dr. Gloriani, you go first.
Okay. Remember that when we're doing gene editing for said, some of these genetic disorders, it is very precise. It is done for a specific person. They're looking at mutations in these patients and then correcting those mutations. So I don't see anybody who is not being treated getting that same product or whatever. So there is even no what's this, environmental contamination or other concerns here. As I said, it is a very personalized thing. So for the immune responses, we are actually looking at how to strengthen the immune response in these patients. So, but of course, all of these are being done in animals first, using model animals, and before we even move on to humans. The human trials have been there for many years, and it's just the very stringent requirements for safety and efficacy that we're following. That's why they're not out there in the market to cater to everybody.
Okay, thank you very much. You have any addition, Dr. Lema?
Yeah, I was about to say in regards to this question about how can we assure that the products are safe. That's a valid question. Not necessarily has to be focused on the technology used to obtain the product, because we have examples of new plant varieties that were obtained by spontaneous mutation. I mean, there was not even human intervention. The DNA just broke and repaired by itself in a different way with a different sequence. So a new trait was obtained, and that new trait was useful for agriculture, but then it was harmful. For instance, there was a mutant celery plant, a spontaneous mutant, no human interventions to obtain that mutation. And that mutant celery was selected because it had better response to pest. It was pest resistant. But nevertheless, the same thing that plant pest resistant have been harmful to humans. So the plant was inadvertently introduced in the market, and only afterwards people realized that it was harmful to humans. And there was a spontaneous mutation, a very traditional breeding process. So the risks are more related with the final characteristics of the products and not with the technology that was used to obtain them. So it's a valid question from the general public, to say how regulators can do their work better in terms of not allowing harmful products to be in the market, but that shall be more broadly based on the characteristics of the product and not the technology used to obtain them. Because you may be misleading the regulators if you impose the regulators with very artificial definitions on what can be regulated that are based in the product. You are forcing the regulators to pay a lot of attention on certain products and not to see others with the same level of scrutiny.
Okay, thank you very much, Dr. Lema. I'm going to raise the last question, and this is for Dr. Marcos Weiss. This is from John Albert Caran. How viable do you think the use of genome editing will be on microalgae? Do you have initiatives for microalgae-based biofuel or other important pharmaceuticals or vaccine production, for example? Well, those are for Nina and Dr. Weiss.
Well, for microalgae, definitely, I think that can help to improve productivity further. I think with biofuels, we still have issues to make them really economical. But yeah, I do not have concrete examples for that.
Okay. And for Dr. Nina, do you have genome editing for microalgae used for pharmaceuticals, perhaps? Do you have
None that I am aware of. Sorry.
Okay,
No,
thank
I
you
am
very
not
much.
familiar.
Yeah. Let's pause a while to see the last poll, and then we will listen to a 2-minute closing or messages from our speakers. Let's see the poll first. The 5th poll question. Okay, the risks of gene-edited products are higher than GMOs, which are higher than conventional breeding techniques. Okay, thank you very much. Let's see the response. I think our audience are very knowledgeable already on this topic. They're all getting the correct answers. Okay, that's right. So it's a false. Thank you very much. So at this point, we would like to give a chance for our speakers to give their 2-minute take-home messages other than the ones that you have presented, or you would like to emphasize a little bit more on your take-home messages. Thank you. We start with Dr. Lema.
Well, thank you. I have repeated myself many times during the seminar, so I would like to talk a little bit about something different, something new, which is the off-target effects. I think one of the questions that you said was responding in a way, but perhaps the interested person was have a question about off-target effect, and in regards to that. It's important because the public hears about genome editing and off-target effects that we describe that these technologies are being improved to avoid having any edition outside of the target region. Some of these techniques are very effective in doing that. And of course, you have a very different world when it comes to health application versus agriculture or industrial applications, because when you have agricultural or industrial applications, you do genome editing on a plant or a microorganisms. You hope to obtain exactly the edition that you wanted to do, but then you screen different organisms, the different individuals, and you can make a whole genome determination, for instance, and select those that have the edition that you want and no other edition, and then take that to the regulatory offices and ultimately to the market. So the techniques are very precise, and also there is a process where you can discard the plants or the microorganisms that didn't resolve the way you wanted to. In regards to health applications, you only can apply the process once to the patient or something like that. So you don't have much, as well as in agricultural applications in animals, where it's not desirable to have off-target effects even after start, because you don't want to discard the animals that were not edited exactly as you wanted. So when it comes to human and animal applications, there is a lot of energy and a lot of efforts being put on having no off-target effects of this technology.
Thank you, Dr. Lema. Let's hear now from Dr. Gloriani.
Okay, yes. Well, I just want to, the key message really for health applications of this is that these are all for the rarer conditions, the intractable conditions, the chronic conditions for which we have no conventional treatment. So that is where all of this gene editing will apply, not to everybody who has an illness. Now, one thing I failed to actually include, because it is not actually gene editing, but just gene detecting, is how we are using CRISPR technology now for COVID-19 diagnostics. And in the US alone, there are 2 companies, one in Cambridge and one in California, that are testing their platforms to have COVID-19 tested in thousands of people in a short period of time. So this CRISPR technology allows them to put in, all these variations of the variants, the genetic variants of these viruses, and have the results in a few hours. So that is not really entirely gene editing, but that is gene detecting. But I'm sure that later on some gene editing will come in because of the variants of these viruses that we are now seeing. So that is all I say.
Thank you very much, Dr. Gloriani. That's very good. Now we go for Dr. Weiss.
Yeah, I risk to repeat myself, but after 25 years of industrial biotech, I'm really enthusiastic that now is the time to move away from these outdated process-centric regulatory frameworks and to move towards a product-centric perspective where the properties of a product are in the center of attention. Techniques are never the solution to our problems alone, and a single technique cannot solve all the problems. But making responsible use of technology is for me crucial. And in that respect, I feel thrilled after this webinar, appreciate it a lot, and look forward that we can go that path further.
Thank you very much. If we're in live audience, we'll give you a big round of applause. Very, very well. Okay, thank you. And now we are going to introduce to you the Dr. Margaret Corembu, who's going to give us the closing message.
Colleagues, ladies and gentlemen, from the entire ISA family, it has been such a great honor hosting you. Such a diverse and accomplished community of practitioners. From the poll results, we are definitely having a very accomplished set of practitioners. We are deeply humbled by your presence, your participation, and just giving us your precious time to share a dose of knowledge on genome editing together. I know for some of us it's late, but we have stayed on until now. Well, as experience has taught us that, the worst experience teaches us the best lesson. In other words, experience is the best teacher, but we also know that the worst experience teach the best lesson. Sharing knowledge on crop biotechnology over the last 22 years from an ISA perspective has taught us that you cannot talk about applications of cutting-edge technologies in agriculture alone without carrying along health, environment, and industrial applications. And we are starting early to bring this on board through these webinars to ensure that when genome editing is up and running, as we have seen, then we'll be talking about the benefits that it is accruing in agriculture, in health, and in environment. So we want to pull together through addressing these global challenges together in a very inclusive version. So in our next webinar series, we'll continue looking at how these applications are running across the three main sectors: health, agriculture, and environment. But most importantly, we'd also like to look at how best to communicate the same processes so that our regulatory systems can become easier. And as Marcos frustratingly keeps repeating, that we hope we are not going to continue going round and round. We need regulations, but we need these regulations that can support innovations and their applications in all these fields. So keep watching, keep with us as we unveil the new ISA. Our next webinar series will be in 2 weeks' time. We'll keep you informed about the topic of discussion, and we'll also reach you out for more on this and others. So please keep up the discussion, keep it lively. Let us continue with those interactions, be part of our network. Thank you so very much.
Thank you so much, Dr. Corembu. And at this point, we would like to thank our speakers for giving us this time to share your knowledge to everybody, to the global community. We have more than 300 participants. And some of the questions were very interesting, but with the limited time, we cannot raise all of them. So they will be provided to you, and then we will be putting together the transcripts of all those answers to those questions. We would like to thank our panelist, Dr. Argentine, who has been answering also some of the questions in the chat group, and also the staff of the Southeast Asia and Afri Center of ISA. Now we'd like to, we're going to invite you to please subscribe to all our materials. So EJ is going to put up there you go. We have the drumbeat, we have the Crop Biotech update, and the Petri dish. And you can subscribe by logging on to your email address. And of course, if you want us to continue this very wonderful webinar, you can go to the ISA.org, donate if you want to donate. Yeah, thank you very much, people. And let's see each other again during the next webinar. Good night, everybody. Thank
Thank
you.
you.
Thank you.
Bye.
Good night.
Right.
Bye.
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 Thank you.
Bye.
Yeah.
Leave.